Okay. Right. So our next speaker is Guillaume, and go ahead whenever you're ready.
Okay, I'm almost done. Okay. So thank you very much for this organizing this very exciting, interesting meeting, despite the difficult conditions. So what I'd like to talk about today is about an old riddle that I have to confess somehow proves that we don't really understand evolutionary biology, but that still isn't an interesting question. So the question is like, can we explain what drives genetic diversity within the species, right? So it's a joint work with my colleague Emmanuel Scherzer. So I put two cartoons. One of them is the Picasso drawing showing that that it's always difficult to change your view on the world. So what you've been taught at school is you think what is reality, but maybe it is not. And the second one relates to time and time distortion, because I think this riddle of what explains genetic diversity has something to do with time scales. So let me, if it does work. Okay, so the framework of this work is the following. So we have some evolutionary process on the left that can be represented like as a diversification process. Say there is a birth rate and a death rate, or whatever models you like. From there is a kinship tree between the organisms, and the organisms are the one we sampled today, which are the color ones here. And we have a kinship tree. The evolutionary process, the tree, cannot be observed, well, unless there is a specific case. What we can observe, though, is a pattern of diversity. So we do have the individuals. Here they are humans, but they could be anything. And they have colors, which are their alleles or their. And from their colors, what we could do is eventually find their kinship trees. So that's the goal of phylogeny. And you can notice that the colors are closer when individuals are closer in their kinship tree. And from the kinship tree, if you are willing to do more, you could eventually go for the evolutionary process that generated that kinship tree. So the goal is like you have the individuals, you have the sequences, for example, and you want to target the evolutionary process. So in the molecular evolution, in the molecular evolution history, there is always nice to have a quick tour of the ancient history. It started maybe with Zucker-Kandel and Pauling in 62, where they demonstrate that the genetic differences scales with divergent time between the species. So if we take sequences in two different species, their amount of mutation they have experienced in their past is linear with time, right? Quickly after that, in 66, Lewotin and Habi dissected the genetic diversity within the species and show that anywhere you look in the genomes, and if you sample a few individuals, you'll find some diversity within the species. Early after that, it was proposed that the vast amount of polymorphism we observed within species are compatible with standard with neutrality, right? So that's Kimura's demonstration. But very quickly also, someone, Richard Leontin, noticed that actually, if you believe the neutral model is correct, diversity should scale with the population size, and actually it does not. So it was stated as like the variation paradox, the Leontin variation paradox, saying like, maybe there is something that we don't capture with the neutral model, because diversity does not scale with population size. So it started like a long debate between what we call the neutralists that are in favor of neutral theory and the selectionists that suspect that selection could actually explain the patterns of diversity. So it's still ongoing, and people have very strong arguments for or against, and see some recent. So the standard neutral model, neutrality, assumes that we have a population, so a species is a population of a constant size, and the size is held constant through time, right? So the vanilla model assumes a constant population size, no structure, so strict panmixia, and no selection, right? Whatever is your type, you have the same distribution of descendants. As a consequence, the number of descendants per individual is Poisson distributed with a mean 1. So two classical incarnation of the neutral models are the Wright-Fisher, the so-called Wright-Fisher model. In a Wright-Fisher model, at each generation, all individuals dies and are replaced by a random sample of the previous generation, right? And you can go on like this. And another incarnation of the same line of thinking is the so-called Moran model, where at each time step, you pick an individual randomly, you kill it, and you replace it by the infant of another individual, right? So in both models, what you do have, so in the Moran model, if you want to scale it at the same time scale the Wright-Fisher, you have to count a generation as n time steps, because in n time steps, you have n death and n births, same as in the Wright-Fisher. So in both cases, the genetic drift time scales is always on the order of capital N generations. And this is true for all exchangeable models. So what I mean by that is like the time scale at which an individual can have a descendant that invade the whole population will always be at the scale of capital N generations. In these mutation drift paradigms, the standard neutral model, individuals carry genetic materials, and their genetic materials can be characterized by some measures of diversity. So let's say heterozygosity, so the probability of having two different alleles when you pick two chromosomes, or the pairwise differences, or many different measures of diversity. And that diversity is assumed to be at equilibrium between an influx at increased diversity, that would be the mutations, and genetic drift that will remove diversity at the scales of capital N generations. So at equilibrium, the diversity in all standard neutral models always is on the order of the population size n times the mutation rate mu. So here starts the problem. We have the model on the left, where you have a constant population size, and that's a model population. It has a constant population size, strict parameter, and not selection. If you pick two random individuals at any time in the population, say this one and this one, the expected time to find a common ancestor between these two individuals is again capital N generations, right? So if you assume that at each generation, there is a rate mu of mutations, the expected number of differences, that is typically noted pi, should be twice the population size, the mutation rate, right? So you go up and down, so that's 2n times mu. That's what you expect under a Wright-Fisher model. So if it's a Moran model, that's going to be n mu instead of 2n mu, but the order would be the same. So that's the model world. And we have the real world on the green side, on the right. In the real world, anything you put in your model will be wrong, right? So you assume constant population size, there will be some demography with variation in population size. You expect in your model, you have strict panmixia. In the real population, there will be structure or preferential matings or whatever. You expect in your model to have no selection, but in reality, you have selection, right? So what population geneticists have invented is this concept of effective population size. So one way to derive the effective population size is simply to estimate the coalescence time between two individuals in a real population. So you take a real population, you sample two individuals, and you would like to know how many generations you have to go back in time to find their common ancestor. And and you call that NE, effective population size. So obviously you cannot access the real number of generations, you have to go back in time. So what you do is you take the diversity, the observed diversity, that is pi ops, and you divide it by twice the mutation rate, and you get an estimate of the divergence time between the two individuals. So here we have the model world, where N, capital N, is the expected time scale that is predicted by the model, and NE is the observed time scale, that is the one you observed in real individuals. The exercise of comparing both values is quite interesting. Say you take humans. Nowadays, we can say we are about 10 to the 10. If you compute the effective population size using diversity, simply by counting how many differences there are between two homologous chromosomes in the human populations and divide by twice the mutation rate, the order you'll find is 10 to the 4. So the question is then, where is where does the factor of 1 million, the one missing million individual is coming from? Why is there a factor of 1 million between NE and N? So some would say, yeah, that's because demography. So why don't we look at other species? So if you take gorillas, now they are 10 to the 5th, and they're getting less and less as they pass by. Their effective population is 1,000. So again, there is a factor of 100. So the Drosophila melanogaster, the famous fruit fly, I don't know the exact number of individuals, but the effective population size worldwide is about 1 million. I don't know how many flies there are on the world, but I suspect 1 million is the number of flies you find in a single tree during the summer in an orchard in US, right? C. elegans, effective population size, 10 to the 5. Have you seen the size of those little nematodes? How many are there in the world, right? So a plant, Thaliana, 10 to the 5th. And I even encountered what's a ridiculous case where you have a Promacocrinus cargelatensis, this species that is like some kind of animals living all around the Antarctica, and the effective population size is 10. So if you go to bacteria, say E. coli, we learned about E. coli just to talk before, the worldwide effective population size is 10 to the 8. I recently asked Olivier Tenaillon for how many E. coli there is in the world. He says between 10 to the 24 and 20 to the 26. So HAV again and again, and you'll find again always the same species, the same picture. The effective population size has nothing to do with the population size. So why is this? If the standard neutral model would make any sense, we would expect diversity to scale with population size. So that's not a new, as I mentioned, that's not a new observation. So could it be structure? So structure, well, yes and no. Locally, it reduces the effective population size, but worldwide, it tends to inflate the global diversity. So I don't think structure is a correct answer. So demography, if the number of individuals per species would have strong deviations where they go into very, very small size and go back to large sizes, that could be the case. But then you have to assume this would be the case for every single species on Earth. Maybe speciation is an answer. Typically very large population, they would tend to speciate into smaller entities that we call species. So there is a maximum number of individuals per species, maybe. Another possibility is selection through linkage. So there are two kinds of effect of selection through linkage. One of them is for negative selection, that is called background selection. And I'm not going to talk about that today. Another one is genetic draft. So let me explain what I mean by genetic draft. So genetic draft comes from an old idea in similar years than actually the Lewontin variation paradox, where you are following. So this is a genome, and you have a site that is selected for, and that's going to be the red square. And in linkage, I mean physically linked with that on the DNA, there is a neutral locus, right? So in the forward model, if you put some advantage on the red square will start invading the population, right? And it would, by a so-called hitchhiking effect, it will drive the fixation of any genetic material that is physically linked with it, right? So see, the gray is here invading the population, but the advantage is only on the red square. But sometimes there will be recombination, and when there is recombination, the association between the gray circle and the red square is broken, so that in the end, some individuals will carry here the white circle, the white disk instead of the gray disk, right? So this process would work provided that the recombination rate is frequent enough so this can happen during the sweep of the red square. So this is the forward so-called hitchhiking effect. We can also rephrase the same model, but look at it backward, right? So if you have the same genome with a selected locus and a neutral locus with some genetic linkage. So if you look backward and you sample individuals after the selective sweep, here the red circles, they will all descend from that first mutant, beneficial mutant that appears, that is here, right? If you sample at a neutral locus, because of recombination, some lineages would escape the sweep. So I think I made another, yes. So if you look at, so this is the forward sweep, the red sweep here, the frequencies. And here there is the association today between the red and the gray disk. If you look backward, if you follow backward in time, they will all coalesce to that single individual that appear here, except, so we are only following, remember, the genealogy of the gray circle, the gray disk, except for the few that escape by recombination. And they recombine with some other loci outside the sweeping. So that's if you have a single sweep, right? This idea was pushed a bit further by assuming there is no single sweep, but there could be recurrent sweeps, right? So assume that if you have recurrent sweeps happening again and again and again, occurring again and again in the population. If you look at the selected loci, you go back to the last sweep and that's it. If you go a bit further away from the selected loci, what you end up with is the coalescent events here are only driven by the sweeps, right? At any sweeps, you coalesce some lineages. But if you go a bit even further from there, you end up having some events, because the recombination is too strong, that do not affect the coalescent tree, but the nodes of these trees are only driven by the sweep, but not all of them. And actually, you can even, in the limit, retrieve a Kingman coalescent tree that is a neutral tree, but for which the time scale is completely different. So this is a cartoon of a simulation, individual-based, where I simulated 6,000 generations on a population of 10,000. And these are the sweeps, right? So you put some sweeps happening again and again and again. And here are the coalescent tree of the last 10 individuals in the coalescent tree. And C, I've just represented the coalescent of the 4 last individuals. And the coalescent event, they do correspond to the sweeps, right? And it shrinks the time by a strong factor. So the question is like, but what factor does it shrink time? It changes the time scale, even though the coalescent tree looks like a neutral tree. So to compute this model, here we have the forward simulations where you have the recurrent sweeps happening, and you are sampling the neutral locus. So here they are the blue ones, and sometimes they can recommend an escape. So we've dissected completely this model, and this can be modeled by actually deterministic sweep, but where the initial frequency and the final frequency are random variables. They are exponential deviations of with a mean twice the selective advantage. The expected sweep time is this one, and the expected time between the two events are this one. So we also computed the probability of coalescent under this model. So what it happens is like, you go back in time, anytime you have a sweep, you toss a coin with that probability. If it's a success, you coalesce the 2 events. Otherwise, you go back in time to the next coalescent event again and again and again. So that's a geometric series, right? So using that, we reparameterize the model so that we have a finite population, so that the selective advantage is somehow scaled with population size. The mutation rate scales as well with the population rate, the population size. And we have this recommendation to selection ratio. In that case, if you have strong selection, rare beneficial mutations, and weak linkage, you end up with a completely neutral tree for which the time scale is distorted. And the time scale is a power of the population size, n to the power of k with k smaller than 1. So what it does is this. You have here the expected time scale, the effective population size expected under genetic drift. This is a log-log plot. Huh? This is a case where the selection is not very strong, right? So you reduce the effective population size, and the effective population size, again, scale as n to the power of k with k minus than 1. If you increase selection or increase the rate of sweeps, you'll reduce again the effective population size. In that case, for example, even though the population size is 10 to the 8, the effective population size is in the order of 10 to the 5th. So short notice, the triangles are forward simulations. The squares are, the circles are forward simulation. Triangles are backward simulations, and the analytics are the lines. So see here with genetic draft, we have an explanation that may be not the correct one for why diversity does not scale with population size. So what do we observe in data? I have only analyzed quickly two data. One of them is birds. Yes, I'm done. Birds, where diversity is actually scales as n to the power of k with k to smaller than 1 with population size, and pinnipeds also similar pattern. So maybe it makes sense. So concluding remarks, neutral theory predicts that diversity should scale with n. What we've done so far is invented an effective population size that using diversity, we have a tautology here, but I think the secret is lying in this selection with a linkage. So that affect diversity and shrink the time scale. Okay, thank you very much.
All right, I won't forget to clap this time. And so we have questions in the chat. If you have other questions, please don't hesitate to ask. So Luisa Fabretti, the estimation of mutation rate could also be a problem for the discrepancy between n and the effective population size.
It definitely could be, but I don't think the mutation rate would vary, let's say, E. coli. The effective population is 10 to the 8, populations 10 to the 24. I'm not sure the variation of mutation rate I mean, there are some errors, but maybe at most at a factor of 10, not a factor of 10 to the 20, right? So there is something else. I mean, it could certainly explain a factor of 10, but not more. I don't think so.
Okay, next question from Eric van Nijwegen. One would have to assume the true mutation rate in the wild is orders of magnitude low.
No, it was not a question. I was trying to answer the question.
Oh, you were trying to answer the question before.
But I have a question.
Go ahead.
So this, these results of how this is going to scale with population size. So how does the size distribution of the recombination events enter into it?
Yeah, so in this toy model, what we've done is we fixed a distance between the neutral locus and the selected locus. So that's the recombination height. So there is no distribution. So the question could be rephrased as, what's the dependency between the recombination rate? So this is the distance and the selected locus.
But you're going to get neutral mutations everywhere, right? Uniformly along the...
Yes, yes, yes. So I think there is an exponential decay, right? So if you go further away,
there must be some scale that says what is the probability for a mutation to be linked to
no. So the way you can rephrase that is like, assume you know the distance to the selected locus. What would be the effect of having the selected locus further away in your genome, right? I think there is an exponential linked between the distance between the neutral mutation and the selected locus. Yes, but I have to check.
Do you have a reference?
It isn't published yet. So
okay. Thanks.
We are writing the paper. Yes, yes, yes.
Okay, I've got a question, not a question, yes, maybe.
Go for it.
A comment. So you assumed when you compared the scaling of the effective population and made the calculations of the effective population and the real populations, you assume that the evolution, if it's neutral, is really neutral completely. So like a flat adaptation, fitness landscape. However, Kimura himself, he did the same when he actually derived the formula. But however, Kimura himself commented on the critique of the neutrality theory in the following way, that actually he assumed only that a significant fraction of mutations is neutral. Well, very small fraction is positive, probably beneficial. And some fraction, substantial, is actually completely deleterious. And what happens if you take this into account? So it's not completely neutral, but neutral and very deleterious ones.
Okay, so here in the model, most mutations are neutral, right? The selected mutations would affect, the key of this problem is you need only a few numbers of beneficial mutations, but because of linkage, they would affect the whole genome. So the first answer is like, I would totally agree that most mutations are neutral. Only few beneficial mutations will actually drive the diversity of the neutral mutations. As far as the deleterious mutations, they also tend to reduce the expected, the effective population size, but not as strongly as the beneficial ones. Does it answer the question?
There's another question in the chat from Michael Manhart. In your simulations, an E scale is less than linearly with n, but they still track each other rather closely. Exponent was still not that far below 1. But the data on NE and N you showed early on sounded like the two were almost independent.
So in that plot, in that plot, in the first, that's the two first parameters we've tried. Obviously, this exponent is almost 1. This one is 0.75. but you could certainly find, if you increase the effect, the beneficial effect of the mutation, or increase the rate of the mutation, you will reduce again the exponent. So the exponent depends on the parameter space. So an interesting, exciting question would be, can we use data to estimate the parameter of the model? That's an ongoing project.
Okay, yeah, that's a really interesting question. So I think with that, we're out of time. So I'd like to thank everybody again for a wonderful morning session. This afternoon, we have a